Everybody, this is the 10-Minute Tutor, and in this lecture today we're going to be talking about the basics of antibodies, antigens, and the structure of antibodies. So this is my first biochemistry video that I'm going to be making, and there's going to be a ton more in the future. And if you haven't already, be sure to check out our previous videos on physics, chemistry, or any other biology videos that we've uploaded. So without further ado, let's get started. How can we define an antibody? So just by writing the term antibody, it has the term anti and body in it. So an antibody, in my book, is best defined as proteins that recognize and neutralize any foreign substances, including bacteria or viruses. So when we hear antibody, they recognize and neutralize foreign substances, but they are proteins most importantly. So antibodies are proteins, meaning they are made up of amino acids. So they are proteins and they're made up of amino acids. And they work by neutralizing and recognizing. So they neutralize foreign substances. And when we're talking about neutralizing foreign substances, it's really going to be neutralizing and recognizing the antigen. The antigen is actually the substance that is going to be identified by the antibody that can evoke a response. So if you're playing a game of baseball, you can think of the antigen as the baseball and the antibody as the baseball glove, because the baseball the baseball fits perfectly in the baseball glove. They have a very compatible structure to each other. So when we're talking about antibodies and antigens, they have a very specific structure for each other and they're biochemically compatible. So they have much to do with an allergic reaction as well. So when we hear allergic reaction, we almost always first think of immune system. So the immune system must be involved somehow in an allergic reaction, right? Especially for an IgE reaction, which we'll talk about a little bit more in the future, but usually the immune system is involved. So during an allergic reaction, the immune system overreacts to an allergen or an antigen by overproducing antibodies. The antibodies travel to the antigen site and cause a reaction. So for allergic reactions in terms of antigens and antibodies, we can think of something like celiac disease. So the celiac disease, and in this example, for celiac disease, we know that the antigen is actually going to be gluten. The protein, gluten, which will evoke a response in the immune system of somebody who has the particular disease. So that's a pretty easy way to think about antibody and antigen in terms of an allergic reaction, which you may. Okay, so what exactly is an antibody made up of, and what is its general shape? So like I said before, an antibody is made up of proteins. So to be made up of proteins, obviously its smaller component must be a bunch of amino acids. So an antibody in general is made up of a sequence of amino acids. Just going to abbreviate AAs. So it's made up of a sequence of amino acids, and an antibody is in general a Y-shaped structure. So that is the most simplest way to first talk about what an antibody is made up of. But there's a few main parts to it. So an antibody is actually made up of 4 polypeptides, or a sequence of amino acids. So 4 polypeptides. And just ignore the nomenclature for now, we just want to talk about the basics. So they were made up of 2 heavy chains, and also 2 light chains. So these chains are made up in a specific amino acid sequence, with the tip of the Y varying most in sequence. So in order to make this very easy to understand, I'm going to first draw this out. So in dark blue, I'm going to draw the heavy chain. So this is the heavy chain of the antibody right here, and it kind of comes up like this. It bends into a Y shape. That is going to be the heavy chain of the antibody. And then to draw the light chain, I'm going to draw it in a lighter color here. This is the light chain here, which kind of just hangs off to the side, to the tip of the antibody. And like I said before, the chains are made up on a specific amino acid sequence. So when we're talking about the tip of the Y, I meant this is the very variable part of the Y. And in this area, this would be called the variable constant. This is going to be called the variable region, and this region here, which represents the lower part of the antibody, is going to represent the constant region. The variable region and the constant region each have their name because the variable region is really where the antigen binds. The antigen usually binds. So that is why it's the variable region, and also the sequence of amino acids differs significantly in the variable region compared to the constant region because the antigen has to bind there. Okay, so now to talk a little bit more in depth about the two regions, the variable and the constant region of antibody, I'm going to just briefly draw out what exactly I'm going to be talking about. So we're going to start off with the heavy chains, and then we're going to have our light chains in blue. And like I said before, this region right here, the region on top, the black region, or the tip of the Y, is known as the variable region, or the variable site. And the region on bottom is actually known as the constant site. But there's a little bit more to it than just these pieces of information. So the variable site is actually known as the FAB site, or the FAB site, which stands for the fragment antigen binding site. So this is where the antigen binds. So it's called the fragment antigen binding site. So we know that because that's where the antigen binds. And the amino acids in this site, the amino acids differ in their sequence. So they differ in their sequence to accommodate the antigen binding to it. And the region on the bottom here in red is the fragment crystallizable site, known as the F sub C, where up here is the F sub AB. And the FC, or the fragment crystallizable site, has an identical sequence of amino acids in all antibodies of the same isotope. So there's different isotopes of antibodies and all that, but for our purposes, the FC has an identical sequence of amino acids. So identical sequence of amino acids, which is why it's called the constant site. There's really nothing binding to it. Usually things do not bind to it. There could be things like macrophages that bind to it, which you may learn, but for our purposes, for antigens, they usually do not bind to the FC site. Okay, so looking a little bit closer at the FAB region, or the fragment antigen binding region, we can actually see a little bit more to it. So when we were looking before, we had these are going to be the light chain, and this was the heavy chain, this was the heavy chain, and this was the light chain. This is just looking at the FAB region here. And by looking at this region, we can actually see the paratope, or the antigen binding site. So there's something called a paratope, which would actually be something like right here, which would be on the tip of the FAB site. And what the paratope is where the antigen actually binds. And there's a sequence of amino acids in this region that is very similar to the antigen. So in this region, there's a very similar sequence of amino acids. Similar to the antigen. So it's just important to know that the last few sequence of amino acids at the end of the FAB site, there are a specific sequence of amino acids that are actually very chemically compatible with the antigen, and that is like exactly where it binds. I know we said that the antigen binds in the FAB site, but specifically on the FAB site, it's on the tip usually, and there is a sequence of amino acids that are biochemically, because of the sequence of amino acids, and stereochemically compatible to the antigens because of the specific shape that the antigen would fit. It's kind of like a baseball and a baseball glove that fits in a specific shape. So we're going to be making more videos on biochemistry and physics and chemistry and biology, so be sure to like and subscribe to our channel as we upload every week.